Hello guys, today we'll talk about the digestive system. So we're going to start with the oral cavity, then the esophagus, then stomach, small intestine and large intestine. So let's start first up with the lip. Now the lip is a structure that makes actually perfect sense. Even if you have some sort of trouble with remembering the structures, just calm down and think of what you have in front of your face. First off, we have the anterior surface is filled with skin. Now the skin in this of course is going to have the stratified squamous keratinized epithelium, which is just again many layers of keratinocytes, dead keratinocytes in fact in the top, and you can actually see that there's anuclear structures in the top of the stratum corneum. Now of course the whole details of the epidermis are going to be discussed, of skin actually we're going to be discussed in a different video. At this point we're just going to focus on the very easy landmarks for you to identify and to distinguish the lip. So first up we're going to have this typical stratified squamous keratinized epithelium. Then you can actually find out many hair follicles, deep invaginations along the whole epithelium of the along the epithelium of course of the skin, and you're going to see in the end of these the hair follicles. Interestingly, most of actually all the hair follicles are going to have surrounding them these unique glands called sebaceous glands. Now these sebaceous glands are lipid producing glands. In fact, the cell itself is going to be the product. This, by definition, makes it a holocrine production of glands. Now, in this case, this is going to be again a sebium. It's going to produce sebium, which is a lipid-rich component, and this is why, of course, if you don't shower for some time, your hair gets all shiny. This is because of the excessive amount of lipids and excessive amount of sebum along the whole line of the hair follicle. Now, of course, we can also find different types of sweat glands. These are the glands that produce serous fluid. This is again watery secretion that contain a high amount of proteins. Of course, contains high amount of anions, cations generally, and most importantly, they can also contain antibacterial and antibacterial peptides and antibacterial enzymes. These again, we're going to discuss the morphology of these structures specifically in the video of the epidermis. But of course, it's easy to identify a serous gland, and we discussed and we've seen of course in previous videos, and the sebaceous will also have seen also the holocrine secretion, so it's easy for you to identify this loosened wide presence of this gland. Now, exactly beneath all of this structure, we're going to find muscle. The question is, what type of muscle? Smooth, cardiac, or skeletal? Well, because the lip is a movable structure, you can move it yourself, so the answer is very easy. It's skeletal muscle. Specifically, we have the orbicularis ori muscle. This is of course aggregations of a high amount of skeletal muscles along the whole line of the lip. Now, between the anterior surface and the posterior surface, we're going to find the vermilion border. Now, the vermilion border is called in this way because first of all it's red, and this is of course the parts of our lips that are red. In this case, the color of the lips is red simply because there's a very high amount of vessels along the whole line of the lip, the lower part of the lip, of course. This is again in the upper part of the epithelium. This is the epithelium itself has no color. Then there is no color, and the only thing that actually can reflect the color of the vessels, and this is exactly why our lips are red to pink. Now, in the posterior border of the lip, we can actually find the oral mucosa. This is typical because this we're going to find typically in this specific part of the mucosa, the oral mucosa, the stratified squamous non-keratinized epithelium. This is, I've already told you to have this association in your head always. Whenever we think of a wet area, your mind should go to stratified squamous non-keratinized epithelium, and vice versa. If you see stratified squamous non-keratinized epithelium, you're supposed to immediately think this is a wet area. Why? Because again, the keratinization process serves the protection and the prevention of water loss. So these are very easy structures to identify, along with of course the mucus secreting labial glands exactly beneath this oral mucosa, and this is of course, we'll see in a bit, a mucus gland again to visualize and to see how they look like. This is the whole structure of the lip. So again, very logical structure, very easy to understand and very easy to identify. This combination of the two different epithelia, the non-keratinized and the keratinized epithelium, the amount of skeletal fiber, the presence of sebaceous glands, these are all unique features that is for you very easy to identify the structure of the lip. Now let's go to tongue. Now the tongue is a structure that of course contains along its surface unique structures. Now specifically, we call these structures papillae. Now the whole area of the tongue contains different types of papillae, sometimes interchangeably, sometimes unique and only present in this location. For example, The whole area of the tongue, 90% of the area of the tongue, is filled with filiform papillae. What is the filiform papillae? In fact, let's visualize them and talk about them at the same time. So this is the filiform papillae. So this area, this is exactly the parakeratinized epithelium. In fact, what do we call parakeratinized epithelium? Parakeratinized is a term that is used to describe an epithelium that has a thick stratum corneum, but it still contains and still maintains the nuclei on the top layer of the epithelium. Now, these structures are parakeratinized. Specific areas of the tongue are parakeratinized, and some are fully keratinized. Of course, it is easy to distinguish simply because if you look at the structure, you can see nuclei on the top. This is parakeratinized. If you don't, it's fully keratinized. So these projections, these projectile structures, are actually the filiform papillae. What's the function of the filiform papillae? Well, this is again a structure to resist abrasion and to, of course, be able to induce friction. Why do we care so much? Well, because food passes by, and because we need a structure that, of course, allows the food to actually have friction forces along the line of the surface of the tongue, and of course to be able to resist these friction forces. And this is exactly why we have the filiform papillae. Important to understand that the filiform papillae play and serve no other role than just food. They do not contain taste buds, unlike the other structures that we're going to see in the papillae. We have also the foliate papillae, the circumvallate papillae, and the fungiform papillae. Now, the fungiform papillae and the circumvallate papillae are the ones that contain the most amount of taste buds. Specifically, the circumvallate papillae contain the highest amount of taste buds in the oral cavity. Let's actually visualize the structures. This is the typical appearance of the circumvallate papillae. As you can see, this is an exophytic structure. Exophytic means that it projects outwards from the structure, and of course we can actually see that the epithelium is very, very thick and large, and here we can actually find, again, lightly keratinized depending on the location, parakeratinized or non-keratinized, or fully keratinized based on the presence of nuclei on the top. Now, it is important to understand that for example, here this is parakeratinized. We're going to actually see nuclei in the top layer. Now, this is the whole mucosal surface of the circumvallate papillae. Important to notice these structures. Now, these structures are in fact taste buds. Now, the taste buds are structures, of course, that are of neuronal nature, and they have continuous columnar shaped cells that are, of course, the nerves themselves. This is the taste pore. This is the opening which allows food and molecules of food to pass through and, of course, be sampled by these taste buds. And on the base, we're going to find the basal cells. Always, always, always a basal cell has a common function of regeneration and reparation when it comes to this specific structure. For example, in this here, the basal cells are also progenitor cells of the taste bud cells, of the cells of the receptor, of the taste receptors. So this is again the typical appearance of the circumvallate papillae. To talk about the general structure of the tongue just a bit, of course, we have again the mucosa, outlined the mucosa, of course, the lamina propria and the loose connective tissue beneath the mucosa. Then we're going to find, of course, mixture of here we're going to have serous and mucous glands. This specifically are serous glands right here, and a very, very high amount of skeletal muscles. There we go. This is increased striated skeletal muscles, continuous. And of course, these are the typical appearance. This is the typical appearance of the mucous glands. We can see the empty appearance of these cells along with the circular appearance, along with the circular shape of the glands. And this is, of course, the typical appearance of the mucous glands. And lastly, we're going to see the vast amount of skeletal muscle along the whole length of the tongue. It's important to understand that actually the tongue is in fact one of the strongest muscles in the human body. So let's move on and visualize the fungiform papillae. Now, the fungiform papillae are in fact smaller, much smaller than the circumvallate papillae, but they're more numerous. Specifically, we have 8 to 12 circumvallate papillae, unlike the fungiform, which are way more and spread out the whole layer of the tongue, spread out the whole surface of the tongue. Now, the structure again is similar, exactly the same appearance. The difference is that these fungiform papillae contain taste buds, but way less in amount in comparison to the circumvallate. And again, it's very easy to identify where it is located. For example, right here, I don't have to zoom in. You can actually see the lucent area. This is the difference between the typical stratified non-keratinized epithelium, parakeratinized epithelium, and the more lucent taste buds present along the whole lining of the mucosa. So, the last one to discuss, we should talk about the foliate papillae. Now, the foliate papillae are specific papillae that are typically seen in young ages, for example, infants and very young kids. This is because throughout time, because of the increased adhesion, increased friction between the food and the foliate papillae, these foliate papillae start to be disintegrated, and in the end there is nothing left to see. So these actual structures can be visible again in infants and very young kids. So let's move on. Important to notice, important to understand, whenever we talk about taste buds and we talk about just nerves, now these nerves are nothing more than just receptors of specific molecules. For example, we have 5 tastes. We have the salt, sour, bitter, sweet, and umami. Now these 5 tastes are nothing more than just specific proteins or specific ions that activate the specific taste receptor and hence lead to the the gustatory feeling of a specific taste. For example, the salt is nothing more than just an activation of the Na of the sodium activated channels, and as a consequence, this serves the input to the brain, and the brain, of course, we taste the saltiness based on the amount of sodium that is present in the specific receptor. Now, let's talk about tooth. Now, the tooth, we have 3 different specific hard structures, dental structures that we can actually see and discuss, and we have the periodontium. We're going to talk about just a bit about the tooth. Extra information for the tooth for dental medicine should be sought out in oral histology books, for example, Tenkate's or Berkowitz. So, just a quick review of the tooth. First, we have the three different hard dental structures: the enamel, the dentin, and the cementum. Now, the enamel is a structure, specifically this is the hardest structure in the human body. It contains contains 96% of hydroxyapatite. It is organized in specific rods, and of course, the 4% is left of different types of proteins, for example, enamelins or tuftulins. It is important to understand that the enamel itself does not contain collagen type 1, but we do find collagen type 1 along the production and along with the formation of enamel. The cells that are responsible for the formation of enamel are the ameloblasts. Now, the ameloblasts are cells that live only during the production of enamel, and after the completion of this stage, they die and they're never to be seen again in an adult human body. Specifically, this is why we say that enamel is a structure that cannot be regenerated, simply because we have no their ameloblasts are no longer present in the human body. So, exactly beneath the enamel, we're going to find the dentin. The dentin has a pattern of production that is very similar to bone. In what terms? First off, the cell that produces the dentin is called the odontoblast. The odontoblast first produces the pre-dentin, and after a maturation phase reaches the dentin, the mature dentin. Same exact logic is with the bone. The osteoblast produces first osteoid, and after some time matures into the bone matrix that we know. So, of course, the odontoblasts are the structures that are responsible for production of pre-dentin, and after time we have the maturation of the pre-dentin to dentin. Now, it is important to understand that the odontoblasts are specific cells that have one unique feature, that the cellular processes called Thomas's fibers extend from the pulpal base. This is again the pulp. This is the pulpal, let's say, cover the periphery of the pulp, and this is where the odontoblast rests along the whole line of the pulp. Now, the odontoblastic processes reach out and extend specifically for the whole length of the dentinal tubules. And this is, of course, this extension assists along with the process of maturation, along with the process of sensation. And this is important because in this way, whenever we have a fully, let's say, connected cell from the odontoblast to the very end of the dentin, this allows specifically for the odontoblastic reactions to pathology. For example, caries. The odontoblast will react specifically with a specific mechanism you should discuss. Again, this is more special topic, but the point is that the odontoblast can sense the caries and, of course, initiate a defense reaction. Not, of course, always successful, but this is, of course, a reaction that the odontoblast can and is in a position to initiate. Now, then the last structure, dental hard structure we're going to see is the cementum. Now, the cementum is a structure that lines up the whole root, and in fact, there are two different types of cementum: the acellular cementum and the cellular cementum. The difference is that first of all is the location, and second of all, as the name suggests, presence of the cells that produce cementum. Of course, the cementum is produced by the cementoblasts. Now, the cementoblasts are present typically in the apical portion. This is, of course, the location of the cellular cementum, and the rest of the root is covered with acellular cementum. Now, the pulp. The pulp is a very important structure because it contains a high number of vessels, a high number of nerves, and specifically contains, when it comes to terms of cells, fibroblasts, mesenchymal stem cells, which is very important, then different immune cells, for example, mast cells. We have also macrophages, monocytes, and so on and so on. Typically speaking, this is a rule that you should always keep in mind. The more vessels you have in a specific area, the better this area it is to repair itself or to regenerate specific structures. This is why in the beginning, when teeth are being produced and the pulp is more expanded, we have higher potential for regeneration processes and higher potential for reparation phases, let's say. But when life progresses and time passes by, then the secondary dentin, which is the continuous process of production of dentin, again, dentin is produced throughout the whole life, unlike enamel, and this continuous process and production of dentin eventually, of course, shrinks the pulp, and as a consequence, the canals the apical foramen is shortening and closing, and there is less amount of vessels passing from the apical foramen to the pulp, and thus rendering the tooth more, let's say, more vulnerable to pathologies. Now, this is a specific, very nice picture that actually visualizes and specifically illustrates the processes of this dental blast that extend from predentin to the very, very end of the dentinal tubules. And one last thing I should mention about this uniqueness of the unique features of the tooth, we find a specific structure called the junctional epithelium. This is an epithelium that is unique in the terms that it actually binds actively the tooth surface with the gingival portion. Specifically, we have these are 2 to 3 layers of cells that are able to bind, again, the typical structure of the epithelium against a very, very hard structure of the cementum directly from the gingiva to the tooth, and this is called the junctional epithelium. So this structure is going to be more typically seen. This is the demineralized structure of the tooth, and this is the ground section. The ground section, of course, contains all the calcium hydroxyapatite, and this is the demineralized structure where we can actually see one very, very important component. As similar to the bone, the dentin is filled with collagen type 1. This is very important because what you should remember, the function of collagen type 1 in the bone, and of course this provides the bone with flexibility, with specific organization along with parallel lines of the hydroxyapatite crystals, and this renders the structure both resistant and, of course, very, very hard, same as dentin. Of course, we have many, many common features between the dentin production and bone matrix production, and you should keep this in mind. So let's move on to the general structure of the GIT. What does this mean? This means that the whole GIT structures, for example, the esophagus, the stomach, the small intestine, the large intestine, in fact share common features. Specifically, all the layers are exactly the same, though there are specific smaller variations when it comes to the contents of the layers, but the layers themselves are exactly the same. Some contours will change. We'll see exactly what kind of contours will change throughout the whole video. So let's talk a bit about the general structure, and then we should specialize and actually visualize the differences between the different portions of the GIT. So first, we have the mucosa. The mucosa is the layer that covers up the whole lumen. For example, let's talk about the esophagus. Let's talk about the esophagus, for example, just a second. And this is again, first we can find the mucosa. Now the mucosa, there is going to contain 3 different layers. The epithelium, and always I told you, the epithelium, always under the epithelium you will find the subepithelial loose connective tissue. Here it's also called lamina propria. And lastly, we're going to find the muscularis mucosa. This is a very, very short and small layer of smooth muscle cells that actually covers up the whole lamina propria. Exactly beneath the the mucosa, we're going to find the submucosa. This is a specific area that typically contains many blood vessels, and always we have the submucosal nerve plexus, also called Meissner's nerve plexus. Now, the Meissner's nerve plexus serves two functions: to control the epithelium and to control the muscularis mucosa. Now, there are sometimes in the specific chart we're going to find the submucosal glands. Other structures in other areas we're not going to find any glands present in submucosa, but sometimes we will. So, for example, right here in the esophagus, we're going to find some mucosal glands in the last portion of the lower third portion of the esophagus, and they're going to be called cardiac glands. They'll produce mucus. We'll see them in a bit. Now, exactly beneath the submucosa, we're going to find the muscularis. The muscularis is going to contain three different structures, very important. The inner circular layer of smooth muscle cells, and exactly between the inner, then the outer longitudinal layer, which is again smooth muscle cells in the longitudinal section, longitudinal arrangement, and between these two structures we're going to find the myenteric nerve plexus, or also called Auerbach's plexus. So the function of the Auerbach's plexus is to control both of these two layers of the smooth muscles. Why is it important? Well, because the arrangement of the circular and the longitudinal section formed in this way allow for the unique movement of peristalsis, which is a combination of constriction and compression. Compression, constriction, and in this way we allow for the food or for the content of the GIT to pass through from one end to the very other end. And lastly, we're going to find the serosa. Now, the serosa is a layer that is specifically lined up with the mesothelium. We've already seen this type of serosa. Now, in other cases, when in some specific locations, we're going to find that the serosa is not present, and instead is present the adventitia. The adventitia is a structure that is a mixture of loose connective tissue, a mixture of adipocytes, and different blood vessels, and so on and so on. So let's focus up and let's zoom in the esophagus specifically. Now, the esophagus is an organ, as we just know by just general knowledge. We can control the upper part of the esophagus, we can move the upper part of the esophagus, we can and like to swallow, and gradually we lose the voluntary contraction of the esophagus. Just this fact, just this event should tell you that the upper part of the esophagus is filled with skeletal muscle, the middle portion of the esophagus is half skeletal, half smooth muscle, and the lower third is filled with smooth muscle. Now, the epithelium of the esophagus is actually very easy to think and to understand. This is because, of course, we have non-keratinized epithelium in the upper part of the mucosa. This is again because we have this is again a wet area, so there is no point of water loss. So it's very easy to distinguish. Exactly beneath, we're gonna find it. We're gonna find the submucosa. This is again here. In this case, we're gonna find no glands in this case, and we can actually see the the connective tissue. Exactly beneath it, we're gonna find the the smooth muscle cells, the circular, and lastly the longitudinal section of the smooth muscle. Exactly, this is the circular, and this is the longitudinal section. And lastly, we're gonna find the tunica serosa in the very bottom along the line of the esophagus. Now, this section, in fact, serves a dual purpose. This, we can actually see the gastroesophageal junction. This is the border between the esophagus and the gastric mucosa. Now, this is another section of the esophagus we can actually find out, and this section serves two purposes. We can actually see the mucous glands, and we can actually see the skeletal muscles. So, again, the outer part, the mucosa, this is again this stratified squamous non-keratinized epithelium. Let's zoom in, and this is again the full length, the full spectrum of this specific epithelium. Exactly beneath, we're going to find the very small amount of the lamina propria. This is again specific the muscularis mucosa, which is controlled specifically again by the Meissner's plexus. This is where we find the mucous glands. Of course, this is not a very typical presentation and the appearance of the mucous glands, but nonetheless we can easily identify that they're mucus simply because they have a high amount of lucent air structures in the area. Here we can actually, this is a specific finding only in this slide. This is again an aggregation of inflammatory cells. This is specific lymphocytes present. Again, if we zoom in, we can understand the lymphocytes because they have very high amount of nucleus and very small amount of cytoplasm. Exactly beneath, we're going to find the This is the submucosa. We find the submucosa extends from the point of the end of the muscularis mucosa to the very beginning of the muscularis externa. So this is again dense connective irregular tissue, and here we start seeing the smooth muscle cells. And we can again visualize and understand they're smooth muscle cells because we have the typical cigar-shaped nucleus present in the along the nucleus, of course. Now, if we go a bit deeper, we can start seeing mixtures of skeletal muscles. If you cannot see it that easily, don't stress, just look around and you'll see way bigger portions, such as this. Here we can actually see the very extensive amount of skeletal muscle. So just by having this information, to have the knowledge that this section contains both skeletal muscle and smooth muscle, you should know that you are in the middle third of the esophagus. Again, upper third of the esophagus contains only skeletal, middle contains mixture of skeletal and smooth, and the lower third contains, of course, only smooth muscles. So let's move on.